Hi everyone, I'm Benjamin Yang. If you're new to this channel, welcome. If you're not, welcome back. If you're new, I make videos based on my experience as a biology tutor, and I hope that my videos will be able to satisfy your curiosity appetite, whether you're young or old. And with that, let's dive right in. At the time of making this video, the market has taken quite a bit of a beating and everything in the market is dropping, including my favorite biotech stock. Nothing has fundamentally changed and there are no adverse news either. As a long-term investor looking to partake in the genomic revolution, this is just another blip in the radar. For those of you who have taken the opportunity to load up on the stock, welcome to our family. Some of you may be asking if it's too late to buy into the biotech stocks now. I think not. We are still in the early days, but it will be a good opportunity to dollar cost average into these companies with great scientific fundamentals that you believe in. Once the first gene editing clinical trials complete successfully, the companies within this sector can potentially take off into the stratosphere. And with that, let's dive right into today's video. Last week, I talked about sequencing companies being complementary to the gene editing companies. A lot of you want me to talk about bio-nanogenomics. I'd like to start off by giving a big picture idea of this subsector by first talking about Illumina, who's currently at the king's seat. Once you understand the scientific fundamentals of this company, then it'll be easier to talk about those who are plotting to dethrone this king. I currently do not hold any sequencing company stocks. I'm only observing and understanding the technology behind it so that I can decide who I will put my money into. As you watch my future videos, you will see my process and adapt it for yourself. And if you stay till the end of this video, I'll reveal who is next in line in this king seat. The idea of sequencing rests on the basis of DNA replication. If we can somehow observe the process real time, then we will know what is the sequence of the DNA. Making the process observational underlies the technology of these sequencing companies. Even though each of the companies may have a slightly different take, the basis is largely the same. So first, let's talk about the fundamentals of DNA replication. DNA is double-stranded and comprises of nucleotides. Each nucleotide has three components comprising firstly a nitrogenous base, A or C or G or T, followed by a pentose sugar, which is like glucose, but instead of a 6 carbon, there are only 5. And finally, 1 phosphate. In order to replicate DNA, the double-stranded structure has to be separated into single strands. Then, each of the strands acts as a template to direct the synthesis of a new strand. This is famously known as semi-conservative replication. To start the process, there's an enzyme known as primase, which adds a primer to the template strand. At the end of the primer is a nucleotide with a 3' hydroxyl group on the pentose sugar. The 3 refers to the third carbon. As the single strand is exposed, free raw materials known as deoxyribonucleoside triphosphates with the correct nitrogenous bases bind to it through the complementary base pairing method, A to T and C to G. The difference between these raw materials and the nucleotides mentioned earlier is that it contains 2 additional phosphates. So, why the need for 2 additional phosphates? This is because the next enzyme known as DNA polymerase adds another nucleotide by breaking 2 phosphates away. This releases energy to power the bond making between the phosphate attached to the 5th carbon of the raw material with the OH attached to the 3rd carbon of the last nucleotide mentioned earlier. And the process repeats until the length of the DNA is completely replicated. Now that we have described DNA replication, it's time to look at how Illumina applies its technology to identify DNA sequences. The process is super complicated, and I'm hyper-simplifying the details. And because of that, I'm not going to describe the entire process, but will suss out just enough information to help you understand what's going on. When a sample containing a person's DNA arrives, it first has to be broken up so that it is easier to sequence. This is likened to cooking beef stew. You don't want to stuff the whole cow into that oven, right? During this process, the DNA is broken into sizes comprising between 200 to 600 pairs of nucleotides. Why pairs? Because DNA is double-stranded and each nucleotide is base paired with another on the other strand. In addition, a short known sequence engineered by Illumina called adapters are added to each end. Each adapter comprises of 3 unique parts. The first part contains sequences complementary to the primers. These primers are part of the reagents needed for the Illumina system to work. The same primers, when bound, allows new strand synthesis by providing the 3' OH group mentioned in the previous section of this video. The second part comprises index sequences that acts like a barcode. This allows the Illumina analysis software to know that the sequences read belongs to the adapter and not to the sample. Since there are 2 different adapters, one at each end, it marks out what sequences has to be read. The third part of the adapter comprises sequences which are complementary to the single-stranded stretch of nucleotides anchored to a special glass plate called a flow cell. Each flow cell comprises, in turn, several lanes. Each lane has got a lot of these nucleotides divided into 2 types, one for each adapter. After the sample is prepared, the double-stranded DNA is separated into single strands and introduced into the flow cell. There, the single strands will complementary base pair to the adapters. DNA polymerase then creates a complementary strand. The double-stranded structure is separated and a buffer washes the original strand away. The new strand, extending from the stretch of nucleotides fixed to the lane in the flow cell, then bends on itself so that the other adapter complementary base pairs with another stretch of nucleotides nearby. The DNA polymerases then synthesize another strand. This way, 2 new strands are synthesized onto the affixed nucleotides on the flow cell. The processes are repeated over and over, producing a cluster of complementary DNA strands. So, what are these clusters for? Each one of the strands will subsequently be read. Because errors may arise during synthesis of new strands, the clusters allow one to derive the numerical similarities between each read strand. If the similarities exceed 97%, the sequences are then validated and therefore can be reported. Once the clusters are formed, the primers are added and will complementary base pair with the strand. The DNA polymerase will then add the correct nucleotide based on the template strand. However, the raw materials used here are slightly modified. For each nitrogenous base, a unique dye is added. This allows the machine to know what base is added each time. During sequencing, for every 1 nucleotide added, a laser beam will be focused onto the flow cell, and this results in the dye emitting a particular color of light. This color will be detected and converted back into DNA sequence. Now, imagine this happening throughout a lane in the flow cell and then every lane in the flow cell, and suddenly many DNA sequences can be easily read very quickly. At the software end, it is able to intelligently compile all the sequences of each fragment that is read, overlapping the DNA sequences and putting back these sequences together from the first to the final nucleotide sequence. I can see why this company is the king of sequencing. Compared to older sequencing techniques, this is much faster and the machine can automate the whole process. Also, the enzyme is usually the most expensive component of the reagents used. And this type of sequencing only requires one, which is DNA polymerase, so the technique is cheaper than others. Finally, the method is very sticky as you need to order the machine that does the sequencing, that comes with a detector that reads the light and converts it to a base sequence, and together with the software that interprets and aligns the sequences. There's also the flow cell that the sequencing lab has to buy together with the primer and DNA polymerases. You might think that this sounds very expensive, but Illumina has managed to reduce the cost of sequencing of the entire genome down to $1,000, and the race is on to further reduce it in the near future. This further adds to the customer stickiness because if the laboratory were to decide to invest in the Illumina machines, they will make it a workhorse in order to justify the purchase. It is therefore unlikely that they will purchase a competitor machine if they have already set up the sequencing workflow. ArcG has initially held Illumina's stocks back in 2020, but I've since sold all and replaced it with Pacific Biosciences instead. So next week, let's compare the two sequencing technologies to see if the decision is justified. And with that, I thank you for staying with me till the end of this video. You've been awesome, and I'm Benjamin Yang. See you in the next video.